Okay, hey y'all, we're doing our next chapter, which is all about nucleic acids. So nucleic acids are responsible for the structures in our DNA and RNA in our cells. And so we'll primarily be talking about it in terms of our DNA. But do you realize this can be used for a lot of different species and everything like that? But we'll talk mostly in terms of our stuff. You will notice for this chapter, I went ahead and wrote most things in. There's a lot of content that would be more writing, less structure drawing. There are structures, but I decided to go ahead and write in everything so that I could just talk through things, and you guys can pause the videos if you'd like to write in, to try and make the videos a little shorter because there's a lot of stuff this chapter. So overall, the central dogma of molecular biology, so like the central idea, is you take DNA that codes for RNA, and then the RNA then codes to make proteins. So this is really important to remember pretty much throughout this whole chapter, that this is the end goal, is to take DNA and code for the proteins that are and enzymes that our body needs. So DNA and RNA are made out of nucleic acids. So nucleic acids is the name for the polymer of nucleotides. So nucleotides are the monomer. Nucleic acids are the polymer. Just like amino acid was a monomer of a protein, and protein was or peptide if it's short, or protein if it's long, for the polymer chain. So let's go ahead and get into the structure, or the basic structure, of nucleotides. So our monomer of nucleic acids are nucleotides. They have three components to them. In blue, you'll see our N-containing base, nitrogen-containing base. It typically has this ish structure, but you'll see exactly what they are below. This nitrogen-containing base is what's responsible for saying what kind of nucleotide there is. There's a lot less nucleotides than there are amino acids, but the other parts are the same, pretty much, depending on which kind of, if you're talking about DNA or RNA. But in general, the identity of that nucleotide is dependent on that base. Then we also have the sugar backbone. So you have a 5-carbon sugar. You'll see the differences in that in a moment, but for the most part, it's pretty much the same. And then we also have a phosphate group. So we've seen these before in our protein chapters, or our lipid chapters, really. So those are the three major parts. So let's go ahead and talk about the bases. So part 1 up here are the nucleotide bases. So again, these are the bases that are responsible for the identity of the nucleotide. These are either derived from pyrimidine or purine. So pyrimidine is this basic structure here. So you can tell it's not like exactly the same, right? But where the nitrogens are is the important part. It's a 6-membered ring with 2 nitrogens, is a pyrimidine, and it'll have different oxidations. So some of them have 2 ketones, some of them have just 1. So it'll be a little bit different. And then, so these 3 nucleotides would be cytosine, which is this one here, which both DNA and RNA have. Thymine, which is the T, is this structure, and this is only found in DNA. And then uracil, which is U, this structure here, this is only in RNA. So thymine and uracil will kind of just swap with one another in DNA or in RNA. So if it's a T in DNA, it would be a U in RNA. And then we have our purine bases. So these are the 6-membered ring next to a 5-membered ring with nitrogens in these particular orientations. Again, you'll see some oxidation, different things like that at different places, but this is overall the structure. These only have 2. There's adenine, which is the A here, and guanine, which is the G. Both of these are in both DNA and RNA, so we don't have to worry about which one's which. You are going to have access to, or you'll have access to notes again for this exam, so it would be wise to make sure these structures are in your notes. But if you're trying to remember which ones are which type, pyrimidines, the the little, I guess, phrase to remember this is cut the pi. So CUT, we have C, U, and T as our nucleotide bases. And then cut the pi, and if you spell pi with PY, then it's the same as PY here, pyrimidine. And then on the other side, you have PU, as in purine, as gold. So you have A and G are your two there. So you can remember those like that, or just make sure you can reference the chart. And then the second part is going to be our pentose sugars. So it's going to be our 5-membered ring sugars. And the two things to remember about this is that in nucleotides, they're always going to be either ribose or deoxyribose. So ribose has this structure here. It is responsible for, or it is an RNA. So RNA stands for ribonucleic acid, meaning that it's this structure. If you take this structure and remove this highlighted OH so that it has a hydrogen there, so you see how it's got hydrogens on both parts, it's missing one of its oxygens. So it's a deoxyribose minus an oxygen ribose. So that's what DNA stands for, deoxyribonucleic acid. And then additionally, we will talk about numbering a little bit on these sugars. Specifically in nucleic acids, you're going to number them with the number and a prime symbol. So carbon numbers with a prime. So we have, for example, here 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. And where this is important is when we put all of this together and make polymers. The last part of those nucleotides are the phosphates, but we already know about those. They just there's nothing fancy to say about those. They're all the same. So that's the general structure of a nucleotide, and we'll get more in depth in the next video.